in mu, delta, epsilon, whatever. You just get one constant region for the light chain.
What does the L stand for in front of the V?
This stands for L. I'm forgetting. I'm going to have to look up why your textbook denotes that. I'll look.
Left? I'm just kidding. What if it was left?
I'll look. But, right, this makes sense to me because I've got my diversity, I've got my joining for my heavy, and then I've got my, but yeah, I'm going to have to remember what L denotes in this picture. So, I know it's a heavy chain because there's diversity regions. Otherwise, it would be a light chain, and it might be like, oh, well, must be what L means. I don't think so. I need to double check our notation and Kudu. So, for humans, this is pretty cool. We have 38 different versions of variable regions up here that can be mixed and matched. We have 14 diversity regions. We have 6 joining regions. And then we have our typical scattered, well, 5, but then there are the subtypes of IgG as well. Yes?
Do heavy chains always have diversity regions, or is it possible for them to not have that?
They always have diversity
Yeah.
regions.
Okay.
Yes. Sometimes they have 2 diversity regions. Weird things. But if you're a heavy chain, you're always going to have a variable, a diversity, a joining, and then you'll be hooked up to a constant region at the end to make the inner stock. Right, then we got to worry about building the light chains, about building arms. So we're going to mix and match. I got to pick one of 38. I got to glue that on to, so this is actually mouse, but if it were me, I'm going to glue that onto one of these 6. And then we've got to choose who we're going to glue onto. In the beginning, all B cells are always going to put these 2 regions. They don't, pieces are going to be cut out of here, and then they're going to be joined to the whole rest of this. The whole rest of this stays on. And as things happen to the B cell, as it senses different things in the environment, as it binds different cytokines, things like this, it will then reactivate rearrangement genes and chop out the intervening sequence to make whatever antibody is necessary. In the beginning, though, you always make these 2. All of the BCRs stuck in the surface are either IgM or IgD. How they get influenced by the environment later on will influence what other types of antibody make. So here is an example of light chain rearrangement. And your book or text, the examples here in slides, we look a little bit at mouse versus human light chain rearrangement because there's slight differences that we figured out after studying this for a while. Mice tend to have a preference for using more kappa chains than using lambda chains, and that's not the way it works out in humans. But the same thing is going on. You have a bunch of variable regions. You're going to add that onto a joining region, and then you're going to plug that onto, in this case, this would be a kappa constant region. So there's only one to choose from. So when you're a light chain, there's only one constant region. You're either a kappa light chain because you rearranged the kappa segment on that chromosome, or you're a lambda light chain because you rearranged the lambda segments and you've hooked up to lambda. Right? So is there a picture of lambda? There is a picture of lambda. The reason why there's a picture of lambda is because lambda is not set up linearly in like the order, right? So look at our heavy chain, right? All the information that encodes all the variable segments is up here. The diversity regions are in a window, the joining regions, and the constant regions, right? Everything is laid out in the order that you would use it. Same thing goes for kappa, light chains. But when we look at the arrangement, the order, the way the genes are laid out, the way the information is laid out in lambda, ooh, it's not the same. You have some variable regions that are upstream of a joining region and a constant region. Then, weirdly enough, there's another joining region and a constant region. Then, downstream, there's some other variable regions, and then some other constant joining and constant regions. So it's not laid out linearly, orderly. Choices are made differently. The deck is shuffled differently in the lambda chain than it is in the kappa chain. So I got questions. Yes?
So what does the side for the chain lambda 4 represent?
Where are we at?
Uh,
We're just giving different notations to different regions based on what is encoded within those regions. It's just a different way of demarcating special little differences that are in the joining regions.
Oh.
Yes.
So if you have the light chain and then the heavy chain kind of next to each other,
Yeah.
right? How, and they each undergo their own splicing and reconfiguration, how come they can both find the same antibody?
So you would never have a kappa and a lambda bound on to the same. So I'll explain why you never mix and match kappa or lambda in a second here. But if there are special mechanisms inside the cell that dictate, if you build your heavy chain, now you have to build a light chain. It's time to turn on the genes for rearranging your light chain. If you choose to start rearranging the chromosome that the kappa genes are on, the lambda chromosome gets silenced. So those genes cannot be accessed, they get turned off. So we try to rearrange those kappa genes, we make a transcript from those rearranged kappa genes, we test the transcript with the heavy chain to see if it works. If it doesn't work, that chromosome gets silenced, and we fire up the lambda genes and we try to rearrange those guys, and we make a transcript and we test the transcript to see if it pairs with the heavy chain. So there's this, it's a cool stepwise process. And the part that kind of blows my mind is the information for building these proteins is on completely different chromosomes, right? So you do gene rearrangement, you rearrange your heavy chain gene, you make a transcript from that gene, the transcript goes to the rough ER, because we're building an anchored member protein, right? So that is going to be translated by a ribosome that's stuck in membrane, so I can insert that protein in the membrane. Then you've got a lambda chain someplace else. It's being rearranged, floating around in the nucleus. That gets rearranged. You get your transcript out of that. That transcript then is not membrane bound, so it could be translated in the rough ER, but most likely it's translated in the smooth ER, and somehow magically somebody knows to click these two proteins together to make the functional whole piece that's in there, right? And we know that proteins have special zip coding that dictate where they go and how they get matched up together, but that's always a piece that kind of blows my mind, that different protein subunit information can be encoded on different chromosomes, and still those pieces still magically find each other. They come together to make a holo protein that works, even though the information comes from different places. Right? Inside our cells is really busy. There's a lot going on there. That's cool. So building the chains, whether it's the heavy chain or the light chain, we're mixing and matching pieces. We're shuffling the deck. Right? This way of shuffling the deck is a little more complicated than kappa, because I could decide to put this chunk with this chunk and this chunk. Or I could take these guys and put them on, one of the ones up here. Right? So there's a lot more cutting, there's a lot more pieces of DNA that get cut out and then sewn back together when we're rearranging this deck. Very interestingly, in mice, 95% of the B cells that get made express a heavy chain with a kappa light chain on it. So almost all of the antibody that is produced, almost all the little B cells floating around have a heavy chain kappa chain pair. And that's the way that they almost exclusively make their VCRs. They can make heavy chain lambda combinations, but they just don't that much. For us, it's very different. We're about a 60-40 split or 50-50 split between our B cells. About half of the population have a kappa and about half of the population have a lambda. So we divide our light chains up more evenly than mice do. And probably some of that makes sense based on our evolution, where we live, how we live, what we do, challenges that we face. This slide is really nice because it gives you an idea about how many gene segments exist from the different pools. How many cards are in each deck? How many cards are in each suit in each deck is what we're getting at too in the numbers here on this table. So we have, I'm going to start with heavy chains first. We have somewhere between 30 to 40 functional heavy chain genes. So 40 different versions of variable regions that we can clip on to that heavy chain. We have about 23 different diversity chains to pick from. Right? If you're a math nerd right now, right, I told you that we can make millions of billions of different antibody combinations, right? So right now you can start hitting on some of that math. 40 times 25, right? And this is just the math that we can account for. Joining regions, right? So 6 functional joining regions, and then our 8 different constant regions that can be added on. So that's just some base math right there. And those are pretty big numbers when we put it all together. Now you got to think about that this is made randomly, and it gets paired randomly with a kappa light chain. So in the kappa genes, we have 40-ish variable regions within the kappa, and 5 different joining regions. Notice they don't account for constant regions, is because the constant region is the same. So the constant region is not adding any variability in, it's not necessary for antigen binding. So we kind of forget about that. For lambda, we have 30 different variable regions that we can make on our lambda light chain, and 5 different joining regions, right? So now we're multiplying actually a lot of numbers together to get larger recombination processes, right? So I can make a whole bunch of heavy chains. I'm going to randomly pair those with maybe a whole bunch of kappa genes or maybe a whole bunch of lambda genes. So that accounts for some variability, but we're still not billions. We're not talking billions. So I'm going to show you where the billions come from. Oh, here's another neat thing to point out that helps explain why mice are so skewed for using kappa genes instead of lambda genes. Look at how many variable regions you have in your kappa pool of genes. Look at how many variable regions you have in your lambda pool. Does it make sense? Evolutionarily, right, based on pressure from the outside world, that kappa would probably be a lot more successful rearrangement for you to use. Yes. Right? But ours are fairly evenly balanced between the two light chain capabilities. So that kind of explains a little bit why we make both of those things in equal pools. Okay. So you might be asking yourself, okay, we're going to lay out the chromosome. We're going to look at all the variable regions, diversity and joining regions. We decide to build a heavy chain. I've got to cut out, right? I've got to take the pieces that I want out of the chromosome, sew it back together, and omit, right? Remove the pieces that I don't want. How do I do this? There has to be, right, mechanistically, a way for this to occur. There's a whole set of very special, very important enzymes that are involved in this process. The two most important enzymes that are absolutely necessary for cutting and pasting the chromosomes back together in the order that we want are variable, right, our VDJC or our VJC, for making heavy chains or light chains, are RAG1 and 2. RAG stands for recombinating activating gene 1 and 2. Right? So we're doing recombination because that's what this is called. It's called gene recombination that allows us to build a unique BCR and a unique TCR. So RAG1 and 2 are essential. How do we know this? There are animals, there are people who have mutations in these genes. Guess what happens to them? They can't make B cells and T cells. You're right. If you can't have an adaptive immune system, you die. You're boy in the bubble disease. You die. Right? Not so good. Not so good, right? So unfortunately, that usually is how we figure out something is really important, how a gene is essential. If it's knocked out and somebody dies, we're like, well, that must have been important, and then we can figure out what it was and what it did. There are a whole bunch of other accessory proteins that are also very important in this process. One you definitely know about, DNA ligase, right? DNA ligase is used during DNA replication. You have, right, replication of both strands of DNA. One strand is the leading strand, right, where replication goes no problem because you move in the 5' to 3' direction, easy peasy, fast. But the other strand, you've got to move in the direction that the replication fork is opening up. And so that means you got to wait. DNA polymerase got to wait around for the replication fork to open. So it goes, then it stops, and it falls off. And then fork opens, and it goes, and it stops, right? But you get fragmented DNA replication. You get Okazaki fragments, right? You got to seal those fragments together. DNA ligase does that. So we borrow an enzyme that we use during replication. We actually borrow a bunch of enzymes that are used during DNA replication and repair to help facilitate this process. You have some very important stabilizer proteins. They help unwind DNA, open it up, hold it open in the regions that we want to cut so that we can cut at the right place and rejoin at the right place. Because, right, I can't just cut wherever I want. I could interfere with the information that's encoded. I could mess up triplet coding, right? All sorts of bad things can happen if I don't have the right mechanical pieces there to unfold the DNA, open it up, hold it stable, allow enzymes to get inside there. That's what essentially all of these guys help to do. There are also some special editing enzymes like TDT, which can add in bases. When we open up the DNA and we cut, sometimes we're going to make cuts that are blunt end cuts or overhang cuts or underhang cuts, and we've got to have enzymes that can come in that can fill in the spaces, right? So we need DNA ligase. We need some editing enzymes to come in there and make sure that we're pairing A with T and G with C, and we're filling in where missing information is. This enzyme is actually pretty unique in that it can add bases in an unscripted way. So sometimes if there are cuts, we can add bases in that aren't necessarily encoded for or scripted by where the DNA cut sites are. So this is going to add random nucleotides in to joining regions where we're cutting genes together, splicing our V's and our D's and our J's and our C's. So we're going to have addition of nucleotides that's unscripted. Sometimes weird things are going to happen there, right? Might not work out the way we want it to. But all of these are critically important for cutting, splicing, and sewing the chromosome back together in the order that we want. It's RAG1 and 2, though, that are the main enzymes that are doing the job. So then the next question is, how do they know where to cut? How do you know where the information, the end of a gene is, and where new information begins? How do I know where the end of V is? So that if I cut here, where's the end of D? How am I going to sew V and D together so that they're in the right orientation to keep the information continuous, to keep reading in the right reading frame, right? The right triplet codon group. There has to be some way for the enzymes to know where to cut, because you can't just cut randomly, right? And sew stuff back together. You're going to goof up the information, you're going to goof up the reading frame. There are special signal sequences that precede and are at the ending of the coding information that tell RAG1 and 2 where is a viable place to cut two pieces of information and to clip them back together. There has to be a way for the cell to know where to cut and paste. This tells us. So we have what are called recognition signal sequences, and these follow a pattern. We have a heptamer nonamer pattern that happens with 23 base pairs. This is usually at the end of a piece of coding information. So if we just look over here, here's a variable region in lambda that I want to clip together to a joining region in lambda. So this heptamer nonamer group here, these act as the little signals that tell RAG, like, oh, as we cruise along here, we're going to keep cruising, we're going to keep, here's the heptamer, here's the nonamer, okay, now it's okay for us to cut. So the signal sequences tell the enzymes where it's okay to clip along. So they read along. When they run into the heptamer and the nonamer, they know, oh, I got to slow down, I'm coming to a cut region, I can cut here. And then they're cruising along, they're going to see on the opposite piece, that they want to sew together preceding this genic information. We're going to have, it's palindromic, we're going to have the nonamer and then the heptamer, right? So this and this are the same. This and this are the same, right? They're going to be mirror images opposite on either side of the two pieces. We're going to cut in the middle and we're going to zip them together. And this is what happens at every variable segment, at every joining segment. It also happens at the diversity segments as well, so that we know which ones, right? There's always going to be a thing at the beginning of the gene that says, here's where you cut, a thing at the end of the gene that says, here's where you cut, so that we know where we can clip the information together. So RAG1 and 2 cruise along, they look for the recognition signal sequences, and they know, oh, here's the end of the information, here's the beginning of that information, chop, sew it together. Ligate it. Now we've clipped it together. Right, so that's just showing us, right, there's a beacon at either side. It tells us what we can cut out, where we can cut, and preserve the information that's inside.
I have a question.
Yes?
This is kind of a big picture question. So all of these different sequences, this is what makes up our immunology library.
Yeah.
And somehow all these different pieces that exist inside of us, if we come into contact with the pathogen, somehow they match up, those cut and pasting,
So we're gonna turn this into a real life structure here in a second. I know this is really weird, right? Like we're zoomed in, super fine tuned right now, right? We're looking at like individual DNA bases, and it's like, what the heck does this mean in the real world? We're gonna zoom back out in a second here, and I'm gonna show you how this results in an interesting protein structure that's going to help us recognize unique antigens.
billions of these just cut and pasted.
You got it. Floating around in you right now, right? Mind blown. Yes?
So it's basically cutting at a palindromic sequence, is it all the time?
Yes.
It's a palindromic sequence?
Yes.
Okay.
Pretty much. Yep. Right? Clever. Simple. Right? Kind of neat. So we're going to know where the end and the beginning of regions are based on where the heptamer nonamer is at. All right, now I'm going to ask you another really weird question to think about. This is a molecular biology question, and we're thinking about the structure of DNA. Notice that in between this heptamer nonamer, we have 23 base pairs, and in between this heptamer nonamer, we have 12 base pairs. Does anything, when we think about the structure of DNA, here's my really, can make more light so that everybody can see this. I apologize right now. I've drawn the structure of DNA about 1,000,000 times. I've never gotten better at it. I'm terrible. So, right, you've got this is my very bad DNA, okay? Okay, so there's my double helix, right? Now I know my picture's terrible, but do we remember anything about repeating structural units? DNA is a staircase, right? Whee, right, all the way around. There's something special about 23 and 12, or one single turn, right? One rung, one, right? Major groove, minor groove. This is the distance, the periodicity of turning of the DNA coil. What? Right? We're thinking about molecular shape right now. How many rungs of the DNA pattern do we have in one turn, right, versus half a turn? So one turn, one big whole turn is 23, half a turn is 12. Okay. So we're repeatedly cutting at 23 and 12, 23 and 12. Why do we think that that might be important? And I'm going to back up, and we're going to think about this picture for a second. Does DNA ever look like this? Naked and flat, like choo-choo tracks? No way, right? DNA is twisted, and it's coiled, it's wrapped around nucleosomes. Yes?
Would it be possible for you to label the 23 and 12 on there?
So, yeah. So I got, this would be, I think, essentially one whole turn, which is 23 to 24 angstroms. So one whole spiral around versus minor groove right inside here. This is around, it's around 13 to 12 angstroms. Like carbon. Right? So this is a really neat thing to think about. As DNA is wrapped around, do we know how many turns of DNA go around one nucleosome? One. One twist goes around. So we're cutting, when we're cutting at spaces that allow us to actually access one wrap and then the space in between the next adjoining wrap of like nucleosomes and DNA. And so that when we're cutting, we can glue the ladder back together so that the ladder actually goes back together instead of cutting it at a turn and cutting it halfway through another turn and then trying to stick it together. It would not work sterically, right? Just physically putting the pieces together. This blows my mind. It's real like chemistry, it's real bonding, it's like, wow, we cut at where the DNA is wrapped and coiled around nucleosomes and turns so that we can actually get to that DNA so we can clip it back together again. Again, mind blown. So the spacing is important so that we can actually hook the chromosome back together in a way that works. Okay, so this shows, I think, a little bit better about where those recognition signal sequences are. So if we're looking in the V cluster, there's a recognition signal sequence at the end of each coding piece of information. And this is a light chain. We're joining a V to a J, right? No D region. The J's are going to have corresponding recognition signal sequences at the beginning.